diversification, and can predict how good are we at predicting the patterns that we see, and when do we fail. And so this talk has been inspired mostly from a symposium that Rosie Gillespie organized. Now it's over 2 years ago. It sounds like it feels like it was like just yesterday, but there was this symposium that Rosie organized that was basically a broad survey of adaptive radiation on a global scale across a wide range of taxonomic groups. And the idea, I think, of Rosie's was to really like see if there's things that we can compare and contrast across all this wide range of adaptive radiation, and can we draw any conclusions in terms of how adaptive radiation happens. And one of the conclusions from this this symposium, and also from the paper that came out of that, is that adaptive radiations actually are very diversified in terms of their patterns. And it's pretty hard to actually predict how they happen and what are the conditions that would lead to certain patterns. And so I'm kind of taking a similar approach, and like got inspired from that, but instead of focusing on adaptive radiation in general, I'm looking at adaptive radiation specifically on islands. And I think there's so many questions that are still left to answer. And I definitely don't intend to try to answer all of these questions in 15 minutes, but rather what I'm gonna do is take a slightly different approach. And instead of like trying to go over a broad survey of island groups and taxonomic groups, I'm going to focus on a single island system and a single type of organisms. And try to address some of these questions. So what I'm going to do is take you to Galapagos. And so for those of you who haven't been there, this is where they are located. And so if we come closer, they're about located like about 1,000 kilometers off the coast of South America, and they're straddling the equator. And so this is a set image of them. And so they're volcanic in origin. And if you color code the islands by how old they are, you get something like that, where you have the older islands that are located in the southeast of the archipelago, and on the west side, you have the younger islands. And the range of age here is between 3.5 million years old to like a few thousand years old for the youngest volcanoes. And then to give you a sense of the diversity in terms of topography, here's a map showing now the elevational gradient across these islands. And what you can see is that these really bright red, bigger spots are found on the younger islands that are much higher than anything else. So across the rest of the archipelago, you have larger islands, but they are fairly low in elevation. And so what that leads us to is vegetation that looks like that on most of the islands. And so like, even though Galapagos straddles the equator, you might think then maybe you'll have kind of tropical climate there. But for the most part, the islands are really harsh. And because they're isolated from the mainland by quite a bit of a distance, this has really resulted in a limited number of organisms that have been able to colonize and successfully live there. And so what we have is habitat that is reduced in its complexity. And again, to give you a feel for what this looks like on the ground, here are shots or videos from these different habitat types. And so here at the bottom, what we have is like the arid zone on an island. And so like the vegetation there is dry, it barely rains during the year. And so organisms that land there really have a challenging habitat confronted with them. Whereas like if you go in the highlands, what you'll have is a habitat that's wet most of the year. It's like raining a lot more. The vegetation is not really tropical-like in the sense that like it's not super high, but it is definitely wet all the time and very green. And so you have these different types of habitats that are really in short distance, and organisms are have the opportunities basically to adapt to these different contexts. And so this has led to the diversification of some iconic fauna, I hate to break it to you, but like I think Galapagos actually has very boring fauna. If you look at in terms of diversity, right? Like, so there is definitely iconic species like the giant tortoises, but with only 11 species, we can't say really that this is a huge feast in diversity. And likewise, we have iguanas, for example, where we have 4 species. Yes, they look great, but in terms of the diversity of form and morphologies and species, it's rather limited. And even like the most iconic of all, the Darwin's finches are pretty limited as well. So I'm not going to keep reviewing all the fauna of Galapagos. We're actually working on a review that is going to look at that. But today what I'm going to do instead is focus on a particular group. And I'm using the slide here as an exemplar of the malacofauna. So basically the terrestrial land snail or the terrestrial mollusk diversity on Galapagos. And so what we have there in terms of endemic species in Galapagos, we have 2 major groups, 2 major genera that have diversified there. So there's the Nasiotis that I'm going to talk about today, and then there's the Succinids, which are the middle group. And then you have another half dozen of genera that are all grouped together in what we call microsnails. These guys are tiny, and there's a handful of species, and I'm not going to discuss them. I feel that they don't really interact the same way with one another, the same way they do the other species, the larger species do. Just to give you an idea of the scale here, these snails are not ginormous in height here. The biggest Nasiotis, the Nasiotis jacobinus on this picture is about ½ centimeter high. So they're still fairly small. So the microsnails are really tiny. Okay, so if we look at Nasiotis snails, I'm just going to quickly give you a survey of what we know about them. There's almost 70 species that have been described. There's probably way more than that. We're still discovering new species when we go. They're found everywhere on all islands and pretty much at all elevation. And all the species we found so far are single island endemics. You can find up to 11 species together in a site, at a given site. And they're not host specific, but they do partition the habitat in a very specific way, depending on the structure that they like to hang on. And their main predators include land birds mostly and other arthropods that we, basically the predation is not really very well characterized. We're just starting in terms of understanding what eats them. In contrast, the Succinids land snails, here's a picture of them. Their shell is much softer and they vary much less in morphology. I'll show you more pictures in a minute. And so in this group, there's only 4 species that have been described. We don't know how many more there are. Nobody has been really studied them. They're found also everywhere. This is also the striking similarity with Nasiotis, is that you actually find them at the same sites at most elevation. But in contrast, with Nasiotis, usually you find only 1 species at every site, sometimes 2 and sometimes 3, very maximum. But for the most cases, what you see is 1 Succinid species and several species of Nasiotis at a given site. And they don't seem to be associated with anything specific. They're truly, seemingly at least, generalists. And we don't actually know what eats them. So we have this contrast, right? We have Nasiotis. Here you have a better idea of the diversity here in shape and sizes and color on the left. And we have Succinids that have been barely studied, but even from based on Felix work, we know that they don't vary much in shape and size. And so here's the work I'm going to present now, like in side by side, the work we've been doing on Nasiotis and now in Succinid more recently. Okay, so this box here is a summary of our general approach. This is months of work that I'm summarizing from my postdoc, but I don't have the time to go into this, so I'm just going to show you the results. If you have questions about how we did this, I'd be happy to answer those. But basically, this is, we generated RAD-seq data and we reconstructed a phylogeny for these species. And so here what you see is 47 of the species, the Galapagos Nasiotis species, And there's a few things I want to point out. So first, you have the map on the left upper corner, and it shows the different colors of the islands. And so if you look at the different clades on the tree, what you notice is that most of the island clades or the island assemblages form these monophyletic groups. Then the other thing that is noteworthy is that there's basically two big groups. If you go down, like the first split basically in Nasiotis, and the split is between the north east part of the archipelago versus the the northwest versus the southeast part of the archipelago. And overall, what we see is a tendency for species to speciate, to colonize and then speciate within island. And so if we reconstruct actually the the pattern of inter-island colonization, this is a stochastic mapping using Bayesian inference. What we see is here is like you have the thicker lines are basically depicting like the most likely routes for these snails to move. The blue or the dot is like, it's a source versus the red dot are sinks. And so we can track the movement of the species across space. And what we see is that movement is relatively rare, and it follows this progression rule where we have species moving from older islands to younger islands. So in summary for Nasiotis, and I see that like I have to speed up a bit here, so I'm not going to repeat myself here, but we have data that suggests that we have a single island colonization from the mainland. Inter-island colonization is rare, and most island assemblages are monophyletic, and we have this pattern of going from older to younger islands as they colonized the different islands. For Nasiotis, I'm going to show you the same thing, but this is very preliminary. So we used CO1 and RAD-seq data for this work. Now the color code here does not correspond to the different islands. What it corresponds to is the different clades for the CO1 and the RAD-seq. So what I'm trying to show here is that our RAD tree is congruent with the CO1 tree. And so the rest of the discussion, I'm going to focus on the CO1 tree, because the CO1 tree, we actually have data from outside of Galapagos. So that allows us to look at colonization from outside of the islands. So what I'm circling here are all the groups that are not Galapagos on the CO1 tree. So this suggests that all the Galapagos Succinids species are not forming a monophyletic group, and the islands were colonized multiple times from the mainland. So we have here species from Japan, Hawaii, and Brazil that are depicted in Peru. And so I'm breaking down the tree here so that we can look at this more specifically. And so we have the main clades here of Succinids. And so one thing I want to point out here is that if you look within each one of these clades, what you have is this connection with a species with another group from the outside of Galapagos. But then if you look at the in-group within that clade, within Galapagos, we see again this pattern of progression rule where the colonization goes from the older islands to the younger islands. And so I know it's to matter. Just trust me, the pattern is there. So we go from like older islands to younger islands multiple times. So this is really much in contrast with the Nasiotis group, because we have multiple colonization from the mainland, but then it's comparable in the sense that once they do reach Galapagos, then they do proceed across the island chain from the old to the younger islands. And so when you do a stochastic mapping of that, it is much messier than the Nasiotis group because of these multiple colonization events that have happened between the islands, but also from the mainland. And I don't know why this picture is so grainy, but you see, you get the idea, right? So basically you have less of a common path, or I guess like more likely path across the islands. You have a lot of crisscrossing going into the. Okay, so in summary, for Succinids, what we see is at least 3 mainland island colonization events that have generated this assemblage of 4 species, presumably. And there's a lot of inter-island colonization. Most island assemblages are not monophyletic, and most inter-island colonization events occur from older to younger islands. So that does match the Nasiotis snails. We don't actually know how many species there are now, because We started with 4, but now we are going to have to revisit all the morphology based on the information we have of the geographical location of these species and their genomics. And we don't actually know if speciation has happened within these islands because we don't know how many species we have, and if speciation happens within habitat, between islands, and so on. So this is all future work. Okay, so I'm just going to take a couple of minutes to try to draw a few conclusions from this. So this RAD-seq approach has allowed us to really dig into the details of the colonization and speciation sequence of these species. And it reveals very different patterns for these two groups. So we have Nasiotis here again on the left, and we have Succinids on the right, and I don't have an X-ray for that species, but basically Succinids are very similar in their morphology. They do have very specific ecology because they do occur everywhere. They occur in very dry habitat and very humid habitats, and these populations are restricted to these habitats, but we don't see a change in morphology associated with this divergence in these different habitats. Whereas in Nasiotis, you see this pattern where as species have diversified and moved into different habitats, they've taken different shapes and sizes and color. So one thing that we think is happening here is that Nasiotis has a different range of predators and have basically evolved to escape predation using their shells, whereas Succinids have very soft shells, and so we're unclear about how they get away from predation, but it's definitely not using their shell. So if you hold a Succinid, you can basically squash it between your fingers, whereas like the Nasiotis ones, like it requires quite a bit of an effort. So we think that predation has a role to play here, and this is something we will investigate further. Okay, I think this is all the time I got. And so I'm gonna just thank all this team. So this is my team of snail hunters that have actually contributed to the work I presented today. Brand provided us a lot of help in the field. He's actually a faculty in the College of Education. So he trains pre-service teachers and he does a lot of science with us as well. And he comes in the field and does lab work. So he's contributed a lot for this on the Succinid project. And obviously there's a lot of funding that went into this and a lot of agencies that have been supporting our work over the years. And so with that, I hope I have a few minutes to take a few questions.
Thank you so much, Christine. That was a lovely talk. It was really, really fantastic. I loved hearing about the snails. Jaime Chavez, he has a question. Jaime, would you like to unmute yourself?
Do you want me to stop sharing the screen or like, doesn't matter?
I don't know if you have any, I don't know if Jaime wants to discuss any slide in particular.
Because I can't see anyone right now, so I don't like that.
Yeah, thank you everyone. Thank you for your talk, Christine, it was fantastic. I'm just curious about the effect of humans bringing snails into the Galapagos, and to which extent all these multiple colonization events that you report are actually not the result of that. And if that is the case, how, if you remove those from your phylogenetic trees, do you think the pattern of diversification colonization will be different? Thank you.
Yeah, this is a great question. So we know we're pretty certain that the clade, the red clade that, like I showed you, that is from Japan is actually a recent introduction. So that one, just because of like how recent it is and how closely related it is to the outgroup. The other ones we're pretty confident that they're actually endemic snails. First, like morphologically, they've been described as endemic to Galapagos, but also based on the genomics, like the splits are much deeper, and so like it makes I guess like we're more confident that those are actually older radiation from that occurred on Galapagos naturally. And so that still leaves us with 3. If I remove like the one invasive or looking like invasive that is coming from Japan, it still leaves us with 3 independent colonization to Galapagos and subsequent radiation across the archipelago. So there's definitely, I think, on like morphological diversity that hasn't been like captured with the description that are out at this point. Thank you.
Well, if you don't mind, we will move on to the next talk. There still is a question for you in the chat, Christine, so we can address it later, or
yeah, I can actually
write
just
it.
write in the chat, right?
Yeah. Well, again, thank you so much for your talk. It was really fantastic. And now we're going to move from snails to plants. And I was looking at Matthew Knope's bio, and he's an old friend of the Bay Area, right, Matt? Because Matt did his PhD in Stanford, and he also did his master's at the San Francisco University. So Matt seems to be very well acquainted with the Bay Area, and now he's a university professor, an assistant professor at the University of Hawaii in Hilo. And it's amazing that Matt is able to join us at 7 a.m. to give his talk. So Matt, thank you so much for being here, and we are really looking forward to hearing your talk.
Thank you so much, Jose, and Athena both for organizing and hosting this symposium. And thank you everybody for being here, for making the time. It's really an honor to be a part of this. So let me go ahead and share some slides here and I'll get started. Let's see. Okay, Christine did a wonderful job of setting up much of what I'm going to talk about today, and then I want to cover some of the same conceptual issues, but I want to travel from the Galapagos about 7,500 kilometers to the northwest to the center of the North Pacific Basin, and talk a little bit about a plant adaptive radiation that we've been working on. It's a genus by the name of Bidens. The Hawaiian name is Ko'oko'olau. They're in the family Compositae, the daisy family. Oops. But before I get started with the talk, I want to wish everybody a very happy St. Patrick's Day. I hope everybody has the opportunity sometime after the talks today to safely celebrate St. Patrick's Day. Okay, so if we take a look at Hawaii, similar to the Galapagos, we see that the islands are arranged in what we call a linear chronosequence. That is, that if you move from the youngest island in the southeast, which is Hawaii Island, or often referred to as the Big Island. Incidentally, the University of Hawaii at Hilo, where I'm located, is right here. The Big Island sits over a magma hotspot, which is basically thought to be a weak point in the oceanic crust that's stationary. And as the Pacific plate moves in a kind of conveyor belt fashion off to the northwest, you have islands that are progressively older as you move from the Big Island to what we refer to as Maui Nui, which is Maui, Molokai, Lanai, and Ko'olave, and continue on to Oahu, and then ultimately to Kauai, which is where I'm actually giving this talk from, which is where my home is. I'm sitting right here, the edge of Honolulu Bay. We get to an island that's about 5 million years old. So just the main Hawaiian Islands time span of about 5 million years. And somewhat similar to what Christine described for the Galapagos, as the islands get older and as they move off the hotspot, they not only subside under their own weight, but they also erode, and they erode quite readily. And they undergo major landslides. For example, you can see the Nu'uanu debris field here, where massive amounts of the islands have broken off. One of the real landmark books in terms of our understanding of the evolution of the Hawaiian flora and fauna is this book edited by Warren Wagner and Vicky Funk, Hawaiian Biogeography, or Evolution on a Hotspot Archipelago. And one of the things that Rosemary Gillespie pointed out to me in this book is that they basically have this general objective, and I'll just read it here, that the chapters in this collaborative work represent the first attempt to test the idea that independently derived groups of Hawaiian organisms exhibit similar patterns of colonization and differentiation that relate directly to the unique geologic history of this oceanic archipelago. And this was a really powerful idea when this was published in 1995. And that basically what it does is it sets up a priori the expectation for what we expect in terms of evolutionary outcomes based on this geologic history, this very clear linear chronosequence of islands. Let me go ahead. So in terms of animal groups in Hawaii, many groups, in fact, most that have been studied, but not all, follow what Christine was describing for the land snails in the Galapagos. That is a progression rule where basically as new islands arise over the hotspot and become available in terms of suitable habitat, animal lineages are able to disperse and subsequently speciate away from older islands, as well as speciate within those newer islands. And all of these studies here for all of these animal groups in Hawaii all show this progression rule, that is dispersal and cladogenesis down the island chain as new islands arise. The situation with plants, though, in Hawaii is quite a bit muddier. The Hawaiian flora is really exceptional in a lot of ways. Just the Hawaiian flowering plants alone, this is excluding non-flowering plants, are over 1,000 native species, all descended from an estimate of original 259 colonists that made it to Hawaii. However, in all of these lineages that I'm showing you here, and all of the studies that I've cited below, each one, all show extremely limited genetic variation, both within species and among species, despite these lineages that often are incredibly species rich and incredibly ecologically and morphologically divergent. And this is what I like to call the Hawaiian plant paradox. That is, that we often find for many lineages, extreme ecomorphological differentiation despite limited genetic differentiation. And of course, this makes working on the genetics of Hawaiian plants a challenge, and it makes it difficult to test hypotheses such as those put forward by Warren Wagner and Vicky Funk in terms of what we would predict and in terms of progression rule and other things for the evolutionary history and biogeographic history of plants in Hawaii. So that's not to say that all is lost in terms of studying the genetics of plants in Hawaii. I want to focus just for a moment on two lineages that are among the best examples of, including others, of adaptive radiation in the Hawaiian Islands. These are what Jerry Carr, formerly of the University of Hawaii at Manoa, referred to as masters of adaptive radiation. The Hawaiian silver swords that you're likely familiar with on the right, and Hawaiian Bidens that we've been studying on the left. As early as 1998, Bruce Baldwin and Michael Sanderson used genetics to demonstrate that the Hawaiian silver sword radiation colonized Hawaii just about exactly the time that Kauai was over the hotspot. So before the formation of Oahu, Maui Nui, and the Big Island. And subsequently, as I mentioned before, as the Pacific plate drifted away and the hotspot had kind of cool periods and then hot periods and built islands, the Hawaiian silver sword radiation, this really exceptional radiation in terms of species richness, in terms of morphological and ecological diversity, has walked down the island chain as predicted by the progression rule. Hawaiian Bidens, though, had been a little bit trickier, and I'll describe why in the next couple of slides. But our work suggests that they're actually quite a bit younger, that they colonized Hawaii within the last 2 million years. And so that basically puts smack middle of the geologic history of the main Hawaiian Islands. So after the formation of Kauai and the formation of Oahu, but before the formation of Maui Nui and the Big Island. And so then do we expect the progression rule for these lineages like Bidens and others that are kind of late comers to the island chain? So let me introduce you a little bit to some of the exceptional diversity of Hawaiian Bidens. This is just a kind of small sampling of some of the species. There's 19 species endemic to the Hawaiian archipelago, including 8 species, subspecies within those. Just to point out a few, you can see here Bidens mauiensis is this very low-lying, succulent, dry-adapted coastal species on the west side of Maui. And contrast that, for example, with Bidens torta. You can see here with Cliff Morden for scale, can grow as woody trees over 10 meters tall in the Waianae Mountains of West Oahu, et cetera, et cetera. They can be found in pretty much every habitat type in Hawaii, from sea level to over 2,000 meters, and from dry forest to rainforest and everything in between. The Hawaiian Biden species are all hexaploid. While their continental ancestors have variable ploidy levels, including some that are hexaploid, but some that are diploid and some that are tetraploid. The morphological and ecological diversity that we observe in these 19 species in endemic to Hawaii is actually greater than the variation amongst the remaining roughly 200 species of Bidens that are found across 5 continents around the world. And we've grown these species in common garden experiments and greenhouses, and they grow true to form. That is, that you get back the phenotypes that you find in the wild. And so all of this morphological diversity is not the result of phenotypic plasticity, but these are fixed genetic differences amongst these species. The species tend to be single island endemics. Within islands, they tend to be habitat segregated as well. And then if you include the fact that this one species, Bidens cosmoides, which is actually right up the top of the mountain here, the top of Kauai and Kokei State Park, is the only species that's bird pollinated. You can see it has a very different floral architecture from the rest of the other 18 species that are insect pollinated. So if you include differences in pollination syndrome, 93% of the species are essentially isolated genetically from one another. However, when they do come into contact with each other at regions of range overlap, hybrid swarms are common. And I just want to point out, even though it's not the focus of this talk and I don't have time to get into it today, that the IUCN Red List estimates that 45% of these species are vulnerable, threatened, or endangered, and are of pressing conservation concern. And sadly, this is not something that's unique to Bidens in Hawaii. It's something that's a general pattern for the entire flora of the Hawaiian Islands, as well as the floras of other Pacific islands. Okay, in terms of primary objectives, what I want to talk about today is basically the work that we've been doing to build genomic resources to be able to address these questions in Hawaiian plants, and in particular using Bidens to do so. So first, can we resolve the phylogenetic history and test the progression rule in Hawaii? And second, can we determine the colonization history throughout the Pacific Islands? And I'll get to that at the end of the talk, but Bidens have colonized other island archipelagos elsewhere in Polynesia. Okay, in terms of methods overview, I'm going to do this very quickly. Basically, we spent the last 15 years collecting, vouchering in museums and extracting DNA from now a total of 91 species from throughout Polynesia, around the Pacific Rim, and elsewhere in the world. This is Bidens samvichensis confusa here in Kauai at the edge of Waimea Canyon. This is a critically endangered subspecies. In terms of molecular techniques, as technologies have changed, we've tried to keep up. We started with Sanger sequencing and then moved to Illumina, HiSeq, shotgun sequencing, and most recently have used the PacBio smart cell long read high fidelity sequencing to generate a high quality reference genome. And I'll talk about that in a little bit. And so what I'd like to do now for the rest of the talk is basically walk through these three molecular techniques in order to talk a little bit about what we get in terms of data types, and a little bit about what we get in terms of results from each of the three, in terms of our understanding of the evolutionary and biogeographic history of Bidens. Okay, so first in terms of Sanger sequencing, very briefly here in terms of methods, we originally went after 4 plastid, that is chloroplast markers, non-coding markers, and 2 nuclear markers. All 6 of these markers have been shown in many groups of continental plants to be good for species level resolution of molecular phylogenies for a total of about 5,400 base pairs, originally with 35 species, including all 19 species endemic to Hawaii and select outgroups. I was going to show you the ITS tree, but I'll just tell you that the individual gene trees from the other 5 markers are almost identical in topology. I want to point out here the node that unites all of the Hawaiian plus these 2 Marquesan species, and I'll get to that in a moment, has strong statistical support. And since it's small, I'll just blow it up here. Basically, what we have on the left here are Bayesian posterior probabilities, on the right, maximum likelihood bootstrap values. And so we have strong support for the monophyly of the Hawaiian plus Marquesan species in the Pacific. You probably noticed, though, that the genetic divergence amongst these incredibly different ecologically and morphologically disparate species in Hawaii is almost zilch at ITS. In fact, the maximum genetic distance amongst the Hawaiian species is less than 1%, whereas if you move through the rest of the phylogeny, the maximum genetic distance across all of these 35 species getting to the African Bidens is about 15%. These 2 species collected by Steve Perlman, at the time undescribed, in the Marquesas, nest within the Hawaiian radiation, but because they're in this basal polytomy with the other Hawaiian species, we couldn't tell the direction of colonization between the Marquesas and Hawaii. And I just want to point out that with this taxon sampling, the location of the most recent common ancestor outside of the Pacific was in or around Mexico. Okay, so then we've moved on to next generation sequencing technologies, and we first used the Illumina, Nextera, XT, HiSeq 4000 shotgun sequencing. Say that 10 times fast. And basically, in short, what we did here is sheared the DNA into fragments and sequenced on this HiSeq platform. We mapped the reads to the common sunflower, Helianthus annuus, because at the time, this was the closest reference genome to Bidens within the family Compositae. We did that both for the nuclear genome as well as the organelles. And we pulled out a subset of reads, first full chloroplast genomes, or what we call plastomes, and the full nuclear ribosomal complex. And this, to give you a little bit of overview in terms of the plastome data, the Bidens chloroplast genomes had about a 98% match to Helianthus with about 1,000x coverage. We have very deep sequencing coverage. The chloroplast genomes, the plastomes across all the species that we included from Hawaii, as well as select outgroups, were all exactly the same size, 150,000 base pairs just about, and about a 25,000 base pair inverted repeat in all of them that we removed for subsequent downstream phylogenetic analyses. In terms of the nuclear data, we went after this about 11,000 base pair section of this ribosomal complex that includes ITS, ETS, and other regions that have been shown to be hypervariable and particularly good at resolving species level molecular phylogenies in plants and continental regions. Basically, what I'm showing you here is the phylogenetic hypothesis generated from that data set. And lots of things I wish I had time to go into that are very interesting in this phylogenetic tree, but I want to point out just a couple of things. First is that we now, for the first time, have a fully bifurcating phylogenetic hypothesis for Hawaiian Bidens, and that most nodes, and you can see that here by the dots, all the dots that are shown here in green have either maximum likelihood or Bayesian posterior probabilities, and usually both, that are greater than 80%. So we're getting some pretty good statistical support in terms of the topology of this tree. Okay, if we also use this same data set to do essentially the same analysis that Christine and colleagues did for the Galapagos land snails, we used biogeobears Bayesian inference to look at this issue of the possibility of progression rule. And basically what I want you to do is focus first in the upper left here, which is a simple kind of cartoon diagram of the island chain from Hawaii Island to Maui Nui to Oahu to Kauai, and they're color coded as both the pies and the phylogeny, as well as the species in the phylogeny from the islands that they're found on. And the overall take-home message here is that each of the solid arrows indicates a founder event with cladogenesis. So all 4 of the founder events and 4 of the 6, which are these dashed lines, which are range expansion events. So all 4 founder events and 4 of the 6 range expansion events are down the island chain following the progression rule. I also want to point out that here, after the formation of Maui Nui, that basically the molecular estimates are following just about exactly the estimate that the geologists tell us, that suitable habitat would be available in Maui Nui for colonization and subsequent speciation on Maui Nui. And then the exact same thing that just about 500,000 years ago, when Hawaii Island becomes available as suitable habitat, there's dispersal from Maui and subsequent speciation away on Hawaii Island, and then subsequent speciation within Hawaii Island. The picture up here is a little bit messier, and that's probably actually exactly also what we would expect for a lineage that's arriving, as I mentioned, at about 2 million years, where Kauai and Oahu, the older islands, are already present, where we wouldn't necessarily expect such clear progression rule, but the younger islands of Maui Nui and Hawaii Island are yet to form, and that's exactly what we find. Okay, and lastly, in terms of sequencing technologies, most recently, with work led by Dr. Renee Bellinger, also of UH Hilo. As I mentioned, we've used the PacBio smart cell sequencing technology, and we've generated a reference genome for Bidens hawaiiensis. This is a species endemic to Hawaii Island, very near UH Hilo. And a couple of things. First, we've independently estimated genome size using 2 different techniques. We've used flow cytometry, and this is the tomato with its known, well-known genome size as a size standard. And we find that the hexaploid Bidens hawaiiensis has a very large genome size estimate of about 7.5 gigabases. That is 7.5 billion bases. In terms of a kind of totally independent way of estimating genome size, based on the sequences from the PacBio sequencing reads and a K-mer analysis, we estimate the genome size there to be about 6.5 gigabases. And so we're pretty happy with this, that they're pretty close to each other. In addition, the K-mer analysis shows 1, 2, 3 shoulders, exactly what we would expect for a hexaploid species. That is, that there's 3 monoploid genomes within the haploid state. And we have also conducted a BUSCO analysis. And for those of you who are not familiar with the BUSCO analysis, BUSCO is essentially a way to analyze the completeness of a genome and compare it to others based on single copy ortholog genes. And what we see is that Bidens right here is a highly complete genome. You can see that here by the very small percentage in red, which is missing data. Less than 10% is missing. Here, based on just 2 smart cells, we have pretty good coverage. We have on average about 21x coverage. But we find that there's a very high proportion of the genome that's duplicated and a very high content of transposable elements. Again, exactly what we would predict for a hexaploid species. Okay, and lastly, and I just want to talk a little bit about work that we've been doing in addition to the Hawaiian radiation. This is work that was really spearheaded by Dr. Vicky Funk, who sadly passed away in 2019. Through the heroic efforts of Dr. Vicky Funk, over the past 15 years, we've sampled all 39 of the 41 species that are found in the archipelagos in the South Pacific, but also within Polynesia. For this, we also use that same Sanger sequencing technology, as well as going after the same original 6 molecular markers that I mentioned in the first part of the talk to make the data compatible. This is just a kind of small smattering of some of the diversity, and the species are color-coded based on the archipelagos where they're found. So in terms of results here, we're really excited about this. We just recently published these results. One of the things that's really interesting about this tree is that it moves the most recent common ancestor of the Pacific radiation from Central America to South America. But I'll point out that the statistical confidence at 62% is not as, as resounding as we might hope it would be. And so we hope to continue to look into that issue. Also here, in terms of the direction of colonization between the Hawaiian radiation and the Marquesan plus Society and Austral Islands, the direction of colonization between the Marquesas and Hawaii is still ambiguous. However, the direction of colonization from the Society Islands is clearly nested within the Marquesan radiation, and then ultimately down to the Austral Islands. Okay, in terms of conclusions, I hope that I've shown you, despite the challenges of working on Hawaiian plants, that phylogenomic methods show promise for improved understanding of adaptive radiations in Hawaii and elsewhere. That understanding the geologic history of island radiations and complete or nearly complete taxon sampling is really key, both to generating and then to testing the evolutionary and biogeographic hypotheses. And lastly, that Hawaiian Bidens adaptive radiation appears to have followed the predicted progression rule dynamics alongside Hawaiian silver sword and other lineages. And I think the implications of these findings are that ultimately it should be possible to have flora-wide phylogenomic analyses of native Hawaiian plants, as well as those from all these other hotspot archipelagos that Carrie Shaw and Rosemary Gillespie pointed out in this wonderful synthesis paper in 2016 from around the world. And that will help us determine what is predictable and what is not predictable, speciation and adaptive radiation on islands and how that works. I also just want to mention that Christine, Ken Hayes at the Bishop Museum, and Jonathan Price, myself, and others are putting together a Pacific Islands Evolution and Biogeography group. And the idea here is basically just to facilitate cross-pollination in terms of research and teaching efforts. We want this group to be as inclusive and as positive as possible. And so if you're interested in being a part of that, just shoot me an email and I'll sign you up for the mailing list. And my email is there at knope@hawaii.edu. I thank everybody in my lab who've been immensely helpful for all of this work that I presented, as well as funding sources. And lastly, I just want to mention, for those of you who aren't aware, Warren Wagner and Jun Wen recently put together this special issue dedicated to the memory of Vicky Funk in the journal of Systematics and Evolution. It's a really wonderful collection of papers. It's fully open access and free to anybody who would like to look at those studies. With that, I'll take any questions. Thank you very much.
Thank you so much, Matt. This was a really fantastic talk. Unfortunately, I can't give you any questions because you run for 27 minutes. So
I'm sorry, I apologize.
No, I mean, I think everyone was happy with all the content and everything you had to show. So thank you so much. We will just jump to Anthony now, Anthony Geneva. And if you have any questions for Matt, please write them on the chat, and Matt, please just take a look at the chat if there are any questions for you. And so the last speaker for today, Anthony Geneva, he's an assistant professor at Rutger University. Anthony is beginning his lab, or has begun his lab quite quickly. Before that, he was in Drexel University, and before he was at Harvard, and he did his PhD at Rochester. And Anthony will be talking about genomes of Anoles, and we're going to shift gears to a completely different ocean, or at least from the Pacific to the Atlantic. So Anthony, the floor is yours. Welcome.
Thank you, Jose and Athena, for asking me to speak. And tough act to follow. Matt and Christine did really excellent jobs. Hopefully, I don't get compared too directly to them because I am quite a departure. Like Jose said, I'm talking about irradiation in the West Indies, also a much older radiation of Anolis lizards. So let's dive in. So everybody has one of these sort of biological diversity slides. I like mine to be textbooks because I like to talk about how anoles are a textbook example of the phenomenon that I'm interested in studying. I'm interested in the origins of biological diversity, and adaptation and speciation are the engines of that diversity. These are processes that operate from the micro to macro level, from within populations up to entire species radiations. And as a result, studying adaptive radiations, which are the coupling of adaptation and speciation, can provide, I think, the best available insight of how adaptation and speciation work together to generate biological diversity. So Anolis lizards, as I said, are this iconic adaptive radiation, and they've become, over time, a model system for the study of evolution and development. The group is quite diverse. There's over 400 species. They occupy the West Indies, Central and South America, and a small part of North America in their native range. They have two traits that are shared by virtually every species. I'm not going to talk too much about these traits, but they're ones that are defining of the group. They have adhesive toe pads on the undersides of their toes that they use to grip the surfaces without actually having to expend any physical effort. And they have these brightly colored flaps of skin that are in males and in some females in some species that are used for signaling, but are a cryptic signal that can be deployed and retracted as the animal wants. They occupy a diverse set of sort of macro habitats, but also micro habitats. And the ecomorphs of Anolis lizards, which I'll focus on for most of my talk today, are defined by their adaptation to distinct microhabitats within a forest environment. So this is some silhouettes of a handful of the ecomorphs. There's one more ecomorph that's not listed here, and then one more putative one that we proposed a couple of years ago, but requires additional study. Decades of evolutionary and functional ecology work have studied the morphological syndromes associated with living in different parts of a tree in a forest. The trunk of a tree has a particular form. The crown, the upper canopy of a tree has a very different animal that's often present there. And what we find is that there are a set of traits that are repeatedly present in the animals occupying those particular habitats across the range of anoles, at least within the Greater Antilles, where about 100 species belong to one of these ecomorph classes. These traits include body size, characteristics of the toe pad, their size and the actual physical structures of them. Limb and body dimensions vary consistently among these ecomorph classes. Their use of habitat obviously is how we first sort of encountered these differences, and those are repeatable in ways that are quantifiable. And finally, behavior among these ecomorphs is consistent. Crown giant anoles act like crown giant anoles wherever they occur across the range of Anolis lizards. So the Anolis adaptive radiation in reality is four adaptive radiations. And that's because on each of the 4 islands of the Greater Antilles, Cuba, Jamaica, Hispaniola, and Puerto Rico, you find animals occupying these distinct subsets of the physical environment, either on the small perches of grass and bushes, or up to these crown giant animals that are occupying the sort of canopies of trees and are quite large, orders of magnitude larger than a twig or grass bush anole. On each of these major Greater Antillean islands, there is a lizard fauna that replicates some or all of these ecomorph classes. I'll get to in a minute why that's particularly interesting, but suffice to say right now that the presence of these replicated radiation provides a natural experiment that's been carried forward over evolutionary time independently on each of these 4 islands. And why can I say independently? Well, each ecomorph has evolved independently on multiple islands. It's not a case of a particular ecomorph or ecological specialist dispersing across the islands of the Greater Antilles. Rather, each ecomorph class has arisen on at least 2 islands, and most of the ecomorphs are present on 3 or 4 of the Greater Antillean, 3 or 4 of the Greater Antillean islands. This figure on the left is sufficiently complicated to not be looked at too closely. The important things to note are that there are clades of different ecomorphs. Each different color here is a different ecomorph class. And there are clades of ecomorphs on each of the major Greater Antillean islands. And on Hispaniola and Cuba, ecomorph types have evolved multiple times. So there are multiple instances of a particular ecomorph class evolving independently on the larger of the 2 Greater Antillean islands. I've been talking about these things qualitatively, but we can actually quantify and show quite clearly that there's very obvious morphological, behavioral, and ecological convergence. So one way to visualize that is in the form of a principal components analysis, where you decompose and project all of a number of different traits that we believe are important for distinguishing among ecomorphs, and identify regions of morphological space that are occupied by individual ecomorphs to the exclusion of the others, or at least to some degree to the exclusion. There's some overlap, as you can see here. And again, remember, twig anoles are a group of ecomorphs that arose independently on multiple islands and have converged to this particular area of morphological space. Another way to state this is sort of in contrast to the phylogeny I just showed you, where ecomorphs are present all over the tree. If you were to do sort of a UPGMA distance analysis of the morphological traits that we measure for anoles, you'd find that the particular ecomorph classes are appear to be monophyletic. They're monophyletic with respect to a distance-based analysis where their morphologies are so similar that they appear to be more closely related to one another than they are evolutionarily. In fact, in most of these cases, you'll see that animals from Cuba, Hispaniola, Jamaica, and Puerto Rico are forming this grouping, UPGMA grouping of crown giant anoles, despite deep divergences among them. I've said deep a couple of times. We don't really know exactly how old the anole radiation is, but the sort of consensus that's arising from multiple different phylogenetic analyses is it's somewhere in the neighborhood of 50 million years, maybe somewhat older, it may be somewhat newer. But we're talking about a relatively old radiation, adaptive radiation, particularly in contrast to hotspot island chains, where those are often, in the tens of millions of years at a maximum. We're talking about a much deeper span of evolutionary time that has benefits and downsides. One argument you could make is that, oh, well, it looks like from the phylogeny you showed me that most anole adaptive radiation, the evolution of ecomorphs, was deep in the tree. And so sampling extant populations won't give you very much information about the processes that gave rise to the morphological forms that are present. And I counter that, new evidence from a couple of really stellar researchers has found that there is recent and ongoing convergent evolution occurring in this group, just to different selective pressures. Kristen Winchell has showed in a series of papers that there is convergent morphological evolution associated with a transition from a forest habitat to an urban habitat. And in those same suite of traits that we're interested in that we use to define ecomorph classes. Similarly, Colin Donahue has shown in a couple of papers that in particular, topad evolution is largely predictable by the frequency with which a region that an animal lives is struck by strong hurricanes, such that you can predict topad size as a function of the intensity and frequency of hurricanes rather than phylogeny and rather than even ecomorph class. There's more information in hurricane frequency than there is in ecomorph class in these data. So what this suggests to me is that we're not just stuck with an old radiation where the traits have been fixed in place and are no longer changing. These traits respond to selection in the current day. That opens up a whole additional suite of population genetic analyses we can perform to try to understand the convergence, the molecular basis of convergence, the morphological convergence that we see within this group. One last thing, because I have you all, Kristen and Colin, lead authors of these two papers, are both postdocs, folks who are in departments and looking for colleagues. You couldn't find better colleagues. Hire these two people, please. Okay, so the question that really gets me interested is what is the genomic basis of all of this morphological, behavioral convergence? There are a couple different possibilities, and disappointingly, I'm not going to give you an answer today. I'm going to tell you how I'm working towards getting an answer, but I don't have an answer to this question yet. Possibilities I see them are, there could be evidence of genomic convergence, either the same sites or genes or gene networks or structural variants could give rise to similar morphologies by mutations in similar genes or gene pathways. An alternative is there could be deployment of segregating ancestral variation, repeated deployment of ancestral segregating variation among this group, such that the genetic basis would be similar, but not because of de novo mutations that arose in parallel and then were fixed in populations, but rather ancestral variation that's been maintained along the group. Now, people who are sort of familiar with this idea might immediately be a little bit hesitant to accept it because, wait a second, you just said the radiation's 50 million years old. You really expect me to believe that there's some ancestral segregating variation sticking around in this group. And I counter that with one sort of observation that we have from Anoles. Anoles harbor tons of genetic diversity within species, remarkably high, suggesting that the effective population sizes of these animals is astronomical, very, very high for a vertebrate. And remember that the rate of coalescence is directly proportional to effective population size. And so even in an old radiation, if it's a very high diversity maintaining radiation, you can have ancestral variation segregating over large spanses of evolutionary time and being available for recruitment in lineages as they arise. And then the last possibility, which to some people would be a disappointment, but I think would be pretty remarkable, is that entirely novel molecular evolution has occurred for the formation of all of these wildly convergent morphological traits. That wouldn't be a failure to me to find that result. That'd actually be pretty shocking, that there are so many different genomic pathways with which you can get animals that look this similar. Now, I didn't say what this was. Let me just mention it. These are twig ecomorphs, trunk ground ecomorphs, and grass bush ecomorphs that are distantly related to one another, that span the depth of that 50 million year clade, at least tree, in at least some of these examples, but they are just striking in the degree to which they're similar. I showed you the sort of quantitative data, which is convincing, but the qualitative data just often blows my mind to see an animal that looks so similar to another one that has a distant relationship and is more closely related, particularly in Jamaica. This animal is more closely related to an animal that looks like this than it is to Anolis cybotes, with which it's quite morphologically convergent. All right, so I'm not the first person to address this question. Russ Corbett-Dedig has worked on this question for a little while now, and he did an incredibly careful, incredibly detailed study trying to identify whether or not there's evidence of phenotypic, or whether or not there's evidence of protein level genomic convergence occurring within Anoles. And at least at his first set of analyses, it seems to be not the case, that there is extensive protein, either site-wise divergence or protein evolutionary rate convergence in Anolis lizards, at least these 12 that he sampled, in leading to the distinct ecomorph phenotypes that are present. Now, I always hate doing this because I think this is a fantastic paper, and whenever you're motivating your research, you need to say, here's what we know now, and here's what we want to know next. And so I have not a single bad thing to say about this paper. It's a fantastic paper, and it's so very careful. Graduate students who are in the audience right now, I definitely suggest you read this because they thought very long and hard about how to be sure that any evidence they found for convergence wasn't due to some other factor other than the actual question they were asking. And they found what's effectively a negative result, a lack of convergence at the protein level, but it's not because they didn't do it the right way. They did it exactly the right way. But I want to move on from this work and ask additional questions. And there's a few things that we can do to move forward. One is that previous to this work, there was really only a single reference genome available within Anoles that could be used to align these data. And as a result, this paper kind of had to focus on protein sequence regions because protein coding regions, because the divergence in non-coding regions would be sufficiently distant that you wouldn't be able to reliably align reads to them. Furthermore, there may be there's an ongoing debate about the relative importance of protein coding and regulatory evolution in adaptive evolution generally. And if one of those sides is correct, then looking for proteins may not actually find you your answer if it's mostly regulatory evolution that's responsible for this. And then finally, there are other types of variants other than SNPs. Things like copy number variants, things like structural variants, inversions may play an outsized role in adaptive evolution in Anoles, and you really can't get at that with resequenced genomic data. So I am, and have been for the last couple of years, assembling new Anole genomes. The way I'm approaching that, let me go a little faster here. I feel like I'm going far too slow. I'm assembling Anole genomes because I want to ask these evolutionary genomic questions. And I feel like you need to have a closely related reference genome to be able to reliably ask questions across the entire genomes of the species you're interested in. Genome scans require linkage information and a well-assembled genome is necessary to use that, those approaches. And finally, chromosomal evolutionary dynamics just can't be assessed with resequence data. I just mentioned that a moment ago. So there are a couple of genomes out there. This is Anolis carolinensis. It was done at very, very high expense and generated a very nice genome assembly with an N50 of 150 megabases. Additional genomes that have been done via Illumina short read sequencing are substantially less contiguous. Following up with what Matt used for BUSCO analyses, this is a plot of BUSCO completeness for these 4 genomes. And you can see that carolinensis, which was done for millions of dollars, has a quite complete genome assembly, but the remaining assemblies aren't quite as good. As I said, I've been using Illumina short read data in combination with 2 other technologies. One developed by Dovetail Genomics, which generates reads that span massive regions, massive space across a genome to scaffold the little pieces that are the component parts of a genome assembly. And I'm not going to go into the details of how that works. And also PacBio hi-fi sequencing, just as Matt has been doing, which are very long, very high quality reads, and then using sort of novel assembly software to perform these genome assemblies. I sequenced right now 9 species from across the done de novo assemblies of 9 species across the Anole radiation. These are selected to capture the 8 major Anole clades. If the root is 50 million years, you can see the age of these individual subclades is quite younger. I have at least 1 de novo assembly per subclade, and there in some cases are 2 for these larger clades. And it includes replication of those ecomorphs that I told you about early on. I've got 3 trunk ground Anoles, 3 trunk crown Anoles, and 2 grass bush Anoles within this de novo assembly set. So let's see what we've gotten from doing these assemblies. Remember, here's carolinensis from the BUSCO that I showed you a moment ago, quite complete, not missing very many genes. These are the 3 genomes developed by a short read assembly. They're okay, but they're actually missing a fairly substantial portion of their genome, so they're somewhat incomplete. And here are the additional genomes that we've done. See that I've redone Anolis auratus and Anolis frenatus, and they've improved substantially. And then these additional genomes all have completeness scores that are comparable or exceed that of carolinensis. And these are done for tens of thousands of dollars each, not millions of dollars each in comparison to the original carolinensis assembly. They're also quite complete. Here's the carolinensis assembly on this plot. The axis here is, again, N50, a measure of contiguity. Also plotted here are other existing reptile genome assemblies. The eastern fed lizard exists as a preprint and is actually quite good as well. And as more and more people use these approaches, we're going to see better and better genomes coming out of this type of work. I believe we're getting chromosome scale scaffolds. And one way to sort of assess whether or not that's the case is by measuring physically the size of these karyotypes and then dividing them by the total genome size, you can get an estimate of what you expect each chromosome to be, the size of what you expect each chromosome to be, and then compare that to your scaffolds. And you can see that I have a very high correlation between scaffold size, the actual size of my scaffolds in my assembly, and the estimated size of the chromosomes based on the C-value estimates you would get from just measuring the physical space occupied by a chromosome in a karyotype. So what do we get from all of this? So now we're able to reliably align next-gen data from any Anole species. I can work on any Anole that I'm interested in now just with these 9 reference genomes, and that enables all kinds of molecular evolution work as well as differential expression analyses that should be really interesting to do. Oh man, I'm really over time, I'm sorry. It also allows for lineage-specific estimation of genome evolution, things like copy number variants, gene family evolution, structural variants. So what I hoped was by now, a year and a half ago, that I would have many of these genomes sequenced and I'd be able to tell you some fun results about the genomic basis of adaptation in Anoles. I don't have those data yet because of the pandemic and various other holdups. I have these genomes, which are fantastic, and they are primed to go for us to do the really fun work that I intend to do next. We're going to resequence hundreds of Anole species ultimately, both for individual lineages, but also some population genetics within them. And we'll use our de novo assemblies as a reference. We then plan to do a number of different phylogenomic analyses to try to identify regions that may be responsible for the convergence, the morphological convergence in Anoles. Those include a method called PhyloGWAS and looking at evolutionary rate shifts on individual branches within the tree. We'll also do PopGen, the sort of standard genome scan approaches, but we can also look within populations and look at things like FST to try to identify regions associated with divergence, convergent evolution that's occurring in those contemporary examples that I mentioned at the beginning of the talk. We've got a really nice set of quantitative genetics phenotypic resources already built from crosses that I've done over the last few years. And so we're ready to start looking using quantitative genetics to try to identify regions associated with traits and the Anole adaptive radiation. And then finally, we want to identify candidate genome regions. And that's because we now have CRISPR working in Anoles. We, meaning the community, this is not my work, but work from Doug Menke's lab. They're able to generate CRISPR transformations within Anoles through a really careful microsurgery technique. And as a result, any candidates that we get, we can actually then begin to interrogate at the molecular level, the functional level, to say, like, are these gene candidates associated with the traits that we believe them to be associated with? So I have to close here. I've gone way too long, and my apologies for that. I think we're in a really good space. We've got a great system. We've got good Anole genomes in place, and some really unique opportunities to test functional evolution because we have this set of genotypic and phenotypic resources that we've built over the last few years. So this is the team that I've been working with. There are others not listed here, but these are the major contributors to it. And I hope there's enough time for a question too. Thanks, everybody.